Hey, welcome to our exploration of genetic recombination and fascinating world of bacterial genetics. Today, we will dwell into Robin's Holiday pivotal work on DNA-strand exchange and why this big breakthrough was crucial in our understanding of genetic diversity and evolution. See, in 1964, this geneticist Robin Holliday proposed a groundbreaking mechanism explaining the gene conversion event during a fungal meiosis process. This proposal introduced the concept of holiday junctions, marking a significant milestone in genetics. Let's dive into uh, this is a paper initially published. Let's dive into the genetic mapping, which serves as a species-specific table showing relative recompositions of genes on um, in the bacterial genome. This tool enables us to understand the genetic distance between the markers and predict the recombination event between the genes. Genetic map. Recombination in bacteria is fundamental genetic process fostering DNA repair, genetic diversity, and acquisition of new traits. So repairing DNA damage is crucial in bacteria, achieving through recombination that replaces or repairs defective sequences within the intact genetic material. Bacteria elevate genetic diversity via transformation, conjugation, or transduction, introducing a new genetic elements and fostering beneficial traits within the population. Homologous recombination aligns similar DNA sequences, promoting holiday junction formation. Site-specific recombination, it integrates or excises the genetic elements in the predefined sites. Okay, the enzyme like RECA, recombinase A, mediated recombination events are a good example of this. Holiday model illustrates genetic exchange featuring heterotublex DNA migration, branch migration, and mismatch repair. The step-by-step holiday model demonstrates how okay how the DNA how the chromosome undergoes nicking, okay, strand invasion, migration, and fostering genetic variability through DNA isomerization. See, in Hardy recombination and crossing over between chromosomes results in gene shuffling, increasing genetic variation, a crucial process in the evolution. Recombination in Holiday model underpins a genetic diversity and evolutionary adaptation, shaping a vibrant world of bacterial genetics. See, here I am showing a step-by-step event lead to the holiday junction formation, branch migration, and then how the repair is done. Okay. Of course, these many times have resulted in shuffling the genes, thereby changing the gene order. I myself have an experience, while working with, while trying to validate DNA barcoding, um, where when I was working with the coral sequences, mitogenome of corals, I have seen that the gene order plays a vital role, even though I couldn't find, even sometimes species of different genus will share not even 0.1% variability in the genome. They are that similar. Given Nidarians, they are supposed to, have identical genome. So, what is the special about this channel? Unknowns. What we still do not know in holiday model. This paper um, has three segments. Those who are still interested in working with this model, this paper clearly shows there is a lack of integration, meaning existing tools are specialized for either HR or HGT direction, and there is a need for comprehensive framework that incorporates both the predictors. This could provide a more holistic understanding of interrelation between the homologous recombination and horizontal gene transfer, that is HGT, and their impact on bacterial genome plasticity. The second important thing this paper has addressed is specificity and sensitivity trade-off. That is, the review indicates that the current tools often exhibit trade-off between sensitivity and specificity. Some tools may excel in accurately detecting certain types of recombination event, but lack sensitivity in other scenario. Developing a hybrid approaches that balances sensitivity and accuracy could enhance overall recombination detection. And the third important issue this paper has addressed, that is published in 2022, is that genomic interrelation, meaning the review emphasized the interconnection connectedness between HR and horizontal gene transfer processes on genomic cell influencing each other in terms of frequency and direction. The comprehensive framework with the predictors for both mechanism would contribute to more nuance and complete understanding of bacterial genome evolution. So the second part of unknown in Holiday Junction is this paper. They clearly addressed a question: what is the mechanism behind Holiday Junction branch migration, which is facilitated by ROBB enzyme, ATPase motor protein, involved in homologous recombination, and how does this asymmetrical assembly of RUB contributes to the coordination of H migration in bacteria? If you are burning with these questions, I think this paper is an uh, ideal paper to read. Okay, and thank you for joining me to this journey through world of bacterial genetics. Stay curious and stay fascinated.